and these are proteins, carbohydrates, lipids, and nucleic acids. You will learn the structure, some of the functions of these biomolecules. Now, let's go directly with today's sessions. At the end of this session, you will be able to list what are the major chemical elements of our life, and also you will be expected to explain what is the significance of non-covalent interactions, which is crucial for the structure of biomolecules. And also you will be expected to discuss the properties and role of water. And finally, you will be expected to describe what is an acid-base balance and what is the role of buffers in order to maintain our acid-base balance. These are the outlines of today's sessions. These are common elements of life, important functional groups: water, acid-base balance, and a buffer. And if time permits us, a classification of carbohydrates. Now, first, before dealing with elements of life, let's see what it means biochemistry means. As you know, you are aware of what biology means and chemistry. And biochemistry, in simple ways, it is a chemistry of life. But before 1903, biochemistry is submerged with other disciplines. But the word biochemistry was introduced by a German chemist known as Carl Nuremberg, and he defined as biochemistry in simple ways. He defined biochemistry is the chemistry of life, and life in terms are defined in biochemistry point biochemical reactions and biochemical processes occurring in subcellular organisms at cell level. Way of definition of life in terms of biochemistry point of view. And when we see biochemistry definitions, we do have when you see living organisms, their difference is visible. Okay, their difference is visible, but at molecular level, all of living organisms, they share what? Some common things. All of them are made from biomolecules. Therefore, anything more than a superficial comprehension of life needs a knowledge of biochemistry. And we do have some of the branches of biochemistry, like medical biochemistry, plant biochemistry, animal biochemistry, and so on. And medical biochemistry deals with the chemical base of human body. Biochemistry tries to deal with the diagnosis and pathological conditions of our bodies. And if you are aware of, or if you have a good knowledge of biochemistry, biochemistry helps you to undergo the right diagnosis. And also, it is useful for you to treat your patients in a good manner. And biochemistry also useful for research in medical fields. Then why we study biochemistry? Why we study biochemistry? In order to know some of the skills, which is important to make us. And once you learn biochemistry, you will be expected to understand chemistry as well as structures of biomolecules. Biomolecules are nothing but They are biopolymers. Okay, they are biopolymers. Are nothing but lipids, carbohydrates, proteins, and nucleic acids. Therefore, once you study biochemistry, you will have a good knowledge of structure and function of these biomolecules. How you will understand its structure as well as its functions by determining their mode of action of biomolecules through isolation as well as structural elucidation will able to us to know the chemistry and structures of biomolecules. And the other point is that why we study biochemistry, because once you study biochemistry, you will have good understanding completely all organized biochemical process which occurs in living cells. Therefore, the aim of biochemistry is to understand how chemical reactions occurs in our body at molecular level. For example, the main function of glucagon is what? To increase blood glucose level. And stored in our body, in the liver as well as in skeletal muscles. But liver levels in between meals, okay, when our glucose level becomes decrease, those glycogen which is stored in the liver starts to degrade. Whereas skeletal muscle glycogen will be able to maintain blood glucose level. Muscle glycogen, when it is degraded, can be utilized for themselves, not able to maintain blood glucose level. This is due to the deficiency of the enzyme called glucose 6-phosphate. Muscle lacks glucose 6-phosphatase. Therefore, when glycogen is degraded, the end product will be glucose 6-phosphate, and this glucose is phosphorylated and it is trapped in the cells, will not escape the plasma membrane. Glucose 6-phosphate, the phosphate group can be removed by glucose 6-phosphatase, and this enzyme is found in the liver. And the final end product will be glycogen, will be changed into maintain as. Then you are aware of the function of glucagon, but here in biochemistry, you will be able to learn what is the mechanism of actions of glucagon at molecular level. Do you think skeletal muscle has receptors for glucagon? Can you hear me? Do you think skeletal muscles has receptors for glucagon? Due to the lack of the enzyme glucose 6-phosphatase, because then and the other aim of biochemistry is if you do have good knowledge of biochemistry, okay, if you are the other aim why we study biochemistry, it is easy for you to identify these. For example, you'll identify inborn errors of metabolism. How inborn errors of metabolism can be occur. For example, inborn errors of metabolism is that inborn errors of can you hear me? Can you hear me? Can you hear me? Can you hear me? Samata? Hello? Okay. Sama? አይሰማችሁም?
አሁን ይሰማል። ያቆራርጣል።
ኦኬ ኮኔክሽን መሰለኝ
ያቆራርጣል።
ኦኬ ይቀጥላል። Okay. The other aim and objective of why you study biochemistry is that if you are to have good knowledge of biochemistry, it is easy for you to identify these mechanisms. For example, PKU is one of the classical example of inborn errors of metabolism, and phenylketonuria is caused by the deficiency of the enzyme phenylalanine hydroxylase, and this enzyme is crucial to change phenylalanine into tyrosine. Now the substrate is phenylalanine. This is one form of amino acids. This phenylalanine is changed into tyrosine through the enzyme called phenylalanine hydroxylase. If there's a deficiency of this enzyme at birth, individuals will develop phenylketonuria, and if it is screened at pregnancy time, mental retardation of the fetus will be decreased if appropriate treatments are given for the patient who have deficiency of phenylalanine hydroxylase. The other is, it is easy for you to understand what are proto-oncogenes, oncogenes, and tumor suppressor genes. Proto-oncogenes are normal genes which is important for cell division. But once proton is changed into oncogenes, always they will have what? Uncontrolled cell division, and this is one of the major cause of cancer. Our cells will try to communicate with each other for the benefit of the organism, not for themselves. Whereas cancer cells will always try division for the benefit of their own, not for the organism itself. And the driving you'll have for you to understand different cancers. Now, these are some of the syllabus of biochemistry. And medical biochemistry is, as you know, it's a branch of biochemistry which deals with a biochemical concept of our body and their interactions. And biochemistry also helps us to try to give some information in order to maintain our health. And for human vitality, okay, for human vitality, metabolism is very crucial. And we do have two major metabolisms. These are catabolism and anabolism reactions. These biomolecules, like lipids, okay, carbohydrates, will undergo catabolized for the production of what? ATP. And this ATP can be utilized for our body to do different activities. And this ATP is useful for anabolic reactions in order to form larger macromolecules from simpler ones. And biochemistry is interrelated with different subjects of medicine. A mutual relationship of biochemistry with medicine, and it is related with physiology, pathology, and so on. Now, biochemistry, you will undergo right diagnosis and treating your patients in a good manner. Finally, clinical biochemistry laboratory test is finally, the important aspect of biochemical laboratory test is useful for you in order to undergo screening, diagnosis, prognosis, and treatment of the disorders. For screening means nothing but screening means you are undergoing to check some biochemical tests without the symptom of the disease in a population level. For example, if there's high prevalence of HIV, after some years later, you will expect there will be high incidence of cervical cancer because HIV/AIDS can be transmitted by ancestors, and one of the causative agents of cancer cells, cervical cancer, is through human papilloma virus, and this virus can be transmitted by ancestors. Therefore, if there without the symptom of cervical cancer, if you are undergoing and checking pap smear test, okay, for cervical cancer, you are without the symptom of the disease, you are undergoing screening, okay. But if the patient visits your clinic, for example, if your patient is expected to have acute myocardial infarctions, criteria according to WHO, one is the presence of chest pain, the other is ECG change test, and the third is increment in cardiac biomarkers. Your patient, if it is positive among the, you will confirm the patient is diagnosed as acute myocardial infarction. This is nothing but the diagnosis. Now you are aware of the disease now, and you will give some treatments to your patient, and you will follow up your patient whether he really from that disease or not. That is nothing but it's prognosis of the disease. Therefore, if you do have a good knowledge of biochemistry, it is easy for you to be a good doctor. Now let's go directly with the organization of life. Okay. The base level, we do have atoms. These atoms can make elements, and these elements can form monomers, and these monomers can combine together through covalent bonds. It will form what? Macromolecules. These macromolecules combine together to form supramolecular structures, and then organelles, cells, tissues, and organisms. Now, what are elements? Okay, elements are substances that cannot be broken down into simpler ones. And in nature, we do have 110 different elements. Among these, we need to have what? 25. And among these 25 elements, the living matter of our body is mainly formed from 6 elements. And these are carbon, hydrogen, oxygen, nitrogen, phosphorus, and sulfur. Among these 6 major elements, carbon is the most dominant and the most versatile element for our life. Why carbon is the most abundant in living organisms? Due to the ability to form carbon chain in unlimited manners. And the carbon can form diverse compounds from simpler to complex ones. For example, if you consider CH4, okay, that's methane, has 1 carbon atom, whereas our DNA has what? Thousands of carbon atoms. And this is the structural hierarchy in molecular organization of our cells. Here, what's expected from you is that just a group of cells can make tissues, okay? Biomolecules it is a building block of our cells. Now, this is the monomeric units of biomolecules, like nucleotides. As you know, nucleic acid is one form of biomolecules, and proteins, it's another form of biomolecules. Lipids, another form of biomolecules, and the other is carbohydrates. And these are some of the corresponding building blocks or monomeric units of biomolecules. Now, nucleotides, DNA or RNA, plus amino acids can make what? Proteins through peptide bond. Okay. Sugars like glucose can make what? Polysaccharides, like in the form of glycogen, cellulose, and so on. Now, at the base, at monomeric units, to form macromolecules, the bond which is responsible is covalent bond. It's a very strong bond. Now the base is very strong, but as level of organization is increasing, non-covalent bond is, for example, when DNA is negatively charged due to the presence of phosphate, but this DNA should be kept in the nucleus. And how it is stable? This DNA is negatively charged, and we do have what? Histones, which is rich in basic amino acids like lysine and arginine. And histones are positively charged. That means due to the presence of high amount of basic amino acids, and these positively charged histones are able to contact with what? The DNA. Okay. Histone plus DNA can form what? Chromatin. Now, chromatin is one form of what? Supramolecular complex. And the other example of supramolecular complex is plasma membrane. As you know, proteins, it can be integral or it can be peripheral. Proteins can be found in the plasma membrane. Even plasma membrane also what? Plasma membrane also has carbohydrates, which is found on the outer leaflet of the plasma membrane. You know, plasma membranes are asymmetrical in nature. That means the composition of inner leaflet and outer leaflet of plasma membranes are not as such symmetrical. Only carbohydrates are only found on the outer leaflet of the plasma membrane. These are nothing but they are called supramolecular complex. And these supramolecular complexes are combined together to form what? Organelles and cells. Now, from this, at molecular level or at the base, we do have what? Covalent bond. Whereas the organization is increasing, non-covalent bond is dominant. The presence of non-covalent bond helps to give our cells to do its own functions or to give flexibility of the cell to do their own function. Can you hear me? Hello, can you hear me? Okay. Now, as I mentioned earlier, biomolecules are what? Can you hear me? Okay. Now, these biomolecules it is a building block of what? Our cells. Just as cells can make tissues, biomolecules can make what? Our cells. And biomolecules are nothing but they are compound of carbon with a variety of functional groups. And different organisms can have what? Different kind of biomolecules. But as I mentioned earlier, superficially, if you see different living organisms, their difference is visible. But at molecular level, all of them all of them shares some basic things. All other kinds of molecules are biomolecules are organic, which is made from 6 major elements. These common things can occur in different living organisms. Among these 6 major elements, carbon is what? The most versatile and the most dominant element. And we do have different forms of biomolecules, okay? Like small molecules, like steroid, or like cholesterol, okay? And other form of biomolecules like building blocks, like amino acids, monosaccharides or sugars, polysaccharides, polymers, like nucleic acids, proteins, polypeptides, and so on. Here, among 4 major biomolecules, like nucleic acids, lipids, carbohydrates, and proteins, lipids are unique. Proteins are made from polymer of amino acids. Nucleic acids are formed from polymer of nucleotides. Carbohydrates are like polysaccharides mainly. It is a polymer of what? Simple sugars. But lipids are not strictly biopolymers. A single molecule of cholesterol can act as what? A lipid. Therefore, lipids are not strictly polymers or biopolymers. Lipids are not strictly biopolymers. A single cholesterol molecules can act as what? A lipid. But the rest biomolecules they are polymers in nature. And we do have a few monomers to make larger macromolecules. Carbohydrates, for example, which is made from what? Some common simple sugars. Proteins, which is made from 20 standard amino acids. Nucleic acids, which is made from polymer of what? Nucleotides. Lipids, as you know, lipids are what? Fat-like substance, but not need to be a fatty acid always. For example, cholesterol, free cholesterol, which is not esterified at third carbon, has no fatty acids. But cholesterol belongs to what? Lipids. Therefore, these are a fat-like substance, but not need to be always a fatty acid. But the majority of them has what? A polymer of fatty acids, and its corresponding alcohol can make lipids. But in strict sense, lipids are not always polymers in nature. And how macromolecules are formed? Macromolecules are formed from what? Their corresponding monomeric units. Through anabolism, through anabolic reactions. Whereas macromolecules, when it's catabolized, it will form what? Their corresponding monomeric units. And all catabolism as well as anabolism reaction is done through the help of what? Enzymes. Now, when you see functional groups, okay, functional groups, as I mentioned earlier, carbon is what? The most versatile element. And if a molecule which has carbon, hydrogen, carbon, hydrogen, carbon, hydrogen, this molecule is somewhat less reactive in nature due to the presence of carbon and hydrogen. If this hydrogen is replaced by other form of functional groups, the molecules can be what? React. Then functional group determines what? The chemical as well as the physical properties of the organic molecules. And these are some of the functional groups which is found in biomolecules. For example, the functional group, if hydrogen is replaced by OH, the biomolecule is considered as an alcohol. If there is COH, the compound belongs to aldehyde, like aldose, okay, carbohydrates, which has an aldehyde functional group, belongs to aldose, whereas carbohydrates, which has functional group of ketone, belongs to ketose, like fructose. Okay? And the other is some biomolecules has carboxylic acid in, like COH, which is present in fatty acids, for example. Okay, fatty acid is made from what? hydrocarbon chain with ends with carboxylic acid. And this is an ether linkage. And these are some of the functional groups which is found in biomolecules. What makes all type of cells shares similarities? What are the similarities among all type of cells? As I mentioned earlier, all living organisms, if you look at them superficially, they are what? Different, and their difference is visible. But at molecular level, all of them shares what? Some basic things which is in common. All cells use what? DNA to store what? The genetic information. But this DNA can be accessed if there is what? RNA. As you know, we do have a central dogma of molecular biology or biochemistry, which starts from DNA, is transcribed to form RNA, and this RNA can be translated to form what? Proteins. Now RNA is considered as what? It's a nucleic acid which is useful to access what? The stored information within the DNA. And all cells use what? Proteins as a catalyst. As you know, all enzymes are not proteins, okay? As you know, all enzymes are not proteins due to the presence of what? Ribozymes. Ribozymes are, they are biocatalysts, but they are not protein-based. Rather, it is an RNA-based enzyme. Due to this, all enzymes are not proteins. Or you can say all enzymes are proteins except ribozymes. And what makes similar, common in all types of cells is that all of them, all type of cells use what? Lipids as part of plasma membrane. Even if the composition of lipids may be vary in different type of cells, but all of them use what? Lipids as component of plasma membrane. All cells use what? Carbohydrate as a recognition if it is found in the outer plate of plasma membrane, and all of different type of cells use what? Carbohydrates as source of energy. And if you look at the chemical composition of our body, large amount of our body is covered by water. Okay, 60% of our body is covered by water. Next to water, the most dominant biomolecules, which account 17% of our body weight, is protein. Next to protein, it's lipid, and the other biomolecules which accounts 1.5 is carbohydrates. This is mainly by glycogen, which is stored in our liver as well as our muscles. And let's see what it means water and its role. As you know, water is the ideal compound for our life, and in living cells, the composition of water accounts from 60% up to 90%. And you know, 3/4 of our Earth's surface is covered by water, and this makes the climate warm, because at daytime, when the environment is very hot, water absorbs this heat, and at night time, when the environment is cool, water releases the heat what they absorbed at daytime. And on Earth, water can exist in different forms, like water can exist in gaseous form, liquid form, and solid form. And here, due to the properties of water, biomolecules or macromolecules will assume to attain their own shape. This is totally determined by what? The properties of water. As you know, water is polar. Okay, as you know, water is polar. For example, if you consider plasma membrane, okay, if you consider plasma membrane is mainly formed from what? Phospholipids. And as you know, phospholipids has what? Both polar as well as non-polar part. Due to the properties of polarity and non-polar properties of phospholipids, plasma membrane is a lipid bilayer in structure. how plasma membrane attains this lipid bilayer structure? This is due to the properties of what? Water. Since water is polar, as you know, 60% of our body is covered by water, and water is polar. The polar part of the phospholipids wants to react with what? Water, whereas the non-polar part of the phospholipids buried themselves away from what? Water. That makes the plasma membrane as such a lipid bilayer in structure. This structure is determined by what? The properties of the water. Another example here is that when proteins are synthesized, initially at primary level of organizations, proteins are nothing but it is a linear sequence of amino acids. But to do their own functions, a given protein will undergo what? Foldedness. Now, how a given protein will undergo foldedness? Foldedness of a protein is determined by what? The properties of water. Since water is polar, those amino acids, which is non-polar, are buried themselves on the inner part of the protein, whereas polar amino acids are found on the outer surface of the protein. That means biological macromolecules assume specific shapes in response to what? The chemical and physical properties of water. Therefore, the property of water determines the shape of the macromolecules. And many of the weak interactions within and between biomolecules are strongly influenced by the solvent properties of water. As you know, water is a good solvent. And when you see the distribution of water in our body, okay, 2/3 of our body water is found on intracellular within the cells, whereas the rest, 1/3 of it is found in extracellular fluid, and extracellular fluid can be divided into two. These are interstitial fluid and the fluid which is found in the plasma, which accounts 20% of the extracellular fluids. What is the function of water? As you know, water can have different functions. It can be useful as transport of nutrients, oxygen into our cells. It can regulate our body temperatures, and water also useful for detoxification of different drugs. Water also helps for hydrolysis reaction, and water also protects vital organs. And water is useful for our metabolism. For example, ATP is energy which is utilized by our cells, and water is needed in order to separate what? The ATP, which is stored the energy in the form of the phosphate group. Without water, ATP will not release what? Energy. And when ATP reacts with water, energy can be what? Produced, and ADP plus inorganic molecule will be formed. Here, water is very crucial in order to release what? The energy which is stored in the ATP. And there is variation of water levels based on tissue type, gender type, and age. As you know, males are rich in what? Lean tissue. Okay. If you compare males and females, as you know, females has what? High fat content than their corresponding males. Whereas males has what? Males have more content of lean tissue than females. Lean tissue means it's a type of tissue which is free from fats. The higher lean tissue means more water content. Then, in terms of gender, males have what? More water content than corresponding females. Since females has what? High fat content and less amount of lean tissue than corresponding males. In terms of age, as age increases, as you know, the amount of lean tissue in our body becomes what? Lost. And in and out, for example, if you consider the in and out going of water in our body is balanced. Whereas as age increases, in and out way of lipids are not controlled just like that of water. As age increase, accumulation of lipids becomes what? Increasing. At the same time, our amount of lean tissue becomes what? Decreasing. And in general, as age increase, amount of water will be what? Lost in our body, and the water content in our body becomes what? Very small. Now the fetus is reaching, and it accounts 85%, but as age increase, there is what? Variation in water content, and elder people has what? Low water content than normal adult. When you see the properties of water, as you know, water is made from what? 3 atoms, 1 oxygen and 2 hydrogen atoms, and oxygen, as you know, it is high electronegative atom. Okay, oxygen is high electronegative atom, and water molecule is formed by sharing of electrons, and it is formed by covalent bond. Water is formed by covalent bond, but as I mentioned earlier, oxygen is what? High electronegative atoms. Even if hydrogen and oxygen shares their electrons to form covalent bond, since oxygen is high electronegative atom, it attracts what? The electron which is shared with hydrogen, and that makes hydrogen becomes partial positive and oxygen becomes partial negative. That makes water is formed by polar covalent bond. We can say water is formed by polar covalent bond. Polarity is due to what? Polarity is due to high electronegative atom of oxygen that makes hydrogen becomes partial positive, whereas oxygen becomes what? Partial negative atom. And many water biological functions is mainly due to its chemical structure, that is due to polarity. And water is a polar molecule due to unequal charge distribution, which makes dipole. Polarity of water causes what? Hydrogen bonding. Okay, and water molecules, okay, each and every water molecules at liquid state is held together by hydrogen bonding, and this is due to what? Partial positive hydrogen attracts what? The partial negative oxygen atoms. And as you know, hydrogen bond is a type of non-covalent bond. Okay, individually it is weak, but if it is too many, it becomes what? Very strong. And especially the liquid form of water, the number of hydrogen bond is not limited. As soon as hydrogen bond is broken, at the same time, there is formation of what? Hydrogen bonding, and the bond of hydrogen bond in liquid form of water is it's limitless. Okay, it is limitless. Now we are looking to see the unique properties of water, which is caused by hydrogen bonding. One of them is cohesion. Okay, cohesion is when water molecules sticks to each other. When water molecules sticks to each other, for example, if we fill a beaker with water, okay, since water molecules stick together, and at the top, it will form what? Surface tension, and this surface tension, it helps some living organisms like insects able to move on, float on the water surface. And the other is addition. Addition means it's when a water molecules sticks with what? Other surface. Sticks with other surface. For example, due to the presence of cohesion and addition, if you fill, for example, half of the beaker with water, and if you add straw on it, the water molecules moves upward. And this is due to what? Water molecules sticks to each other. That is the purpose of cohesion. And the other is addition. Water molecules sticks with what? Just the straw. That is called addition. And this able to give what? Capillary action. And this helps the plant to get water from the ground to reach to the top of the tree. due to the properties of water, and this is done by hydrogen bonds. And the other unique property of water, which is caused by hydrogen bonding, is that when water in ice form, it becomes what? Expands. That means, as you know, ice is less dense than what? The liquid form of water. As I mentioned earlier, liquid form of water, the number of hydrogen bond is what? Not limited. Always there is bond formation. Whereas in ice form of water, ice forms what? Stable hydrogen bond. Each molecule is bonded to 4 neighbors, and it will form crystalline lattice structure. That means the number of hydrogen bonds in ice form is limited and stable, whereas the liquid form is, it's not as such stable, and there is always formation of hydrogen bonds. That makes liquid form of water is high dense than ice form of water. That makes ice floats on liquid form of water, and it's the structures of what? Ice form of water, in which there is ordered formation of hydrogen bonds, which is stable, whereas in liquid forms there is no stable hydrogen bonding. The other properties of water, which is caused by hydrogen bonds, is that water helps our body in order to regulate our body temperature. This is due to the presence of high heat specific capacity of water. And the other is unique properties of water, which is caused by polarity. Okay, polarity. Due to the presence of polarity, water can act as a universal solvent. That doesn't mean that water can dissolve lipids. Okay, as you know, like dissolves like. Polar solvents can dissolve what? Polar solutes, whereas non-polar solvents can dissolve non-polar compounds. And water, as you know, it's polar. It can dissolve what? Polar solutes. And this is a summary. Okay. This is the properties of water. Water is a universal solvent. Why? This is due to what? Polarity. Water can have creation and addition property. This is due to hydrogen bonding. Water resists change in temperature. This is due to what? Hydrogen bonding. Water is less dense in ice than liquid form of water. This is due to what? Hydrogen bonding change. Can you hear me? Hello? Okay, let's continue. The next part is acid base and buffer systems. As you know, pH is minus logarithm of hydrogen concentration, and hydrogen it is nothing but it is really a proton. And we do have a pH range which stands from 0 up to 14. As the concentration of hydrogen is increasing, the solution will leads to what? Acidic. And as you know, pH less than 7 is considered as acidic environment, whereas as concentration of hydrogen is low, the pH becomes what? High, and the solution becomes alkaline or basic. When you see our body pH, homeostasis of pH should be tightly controlled in order to do enzymes to do their own functions and other things. Extracellular fluid, when you see the pH, that means atrial pH, the average pH is 7.4, not 7, okay? Here, the atrial pH is 7.4. That means our extracellular fluid or our atrial pH is slightly what? Alkaline, okay? It's not as such neutral. Rather, it is slightly alkaline or basic. And our blood pH ranges from 7.35 up to 7.45. The average is 7.4. And the survival range of our pH is in between 6.8 up to 8. And an individual can develop acidosis if his pH is less than 7.35, and if it is above 7.45, individuals may develop alkalosis. And acids are nothing but, now we are looking to see some of the definition of acids and the base. Acids are nothing but it's hydrogen donors. Hydrogen donors, while bases are hydrogen acceptors. An acid can be, acids and bases can be strong or weak ones. Strong acid or strong base it is a base or an acid which dissociates what? Completely in a solution. Whereas weak acids or bases are, it dissociates partially in solution. Therefore, if it is a weak acid, there will have what? Undissociated acids in a solution. Weak acids like lactic acid, carbonic acid, and so on. A weak acid has its characteristic dissociation constant, which is known as Ka. And Anderson-Hasselbalch equations gives to know us for the relationship between the pH of the solution, the K of an acid, and the extent of its dissociation. We will see later what is the use of Anderson-Hasselbalch equations. Now, before that, let's see an acid and conjugate base pairs. An acid is represented by Ha, okay? A weak acid is represented by Ha, which dissociates partially. It is indicated by 2 arrow, okay? If an acid is strong acid, it will be indicated by what? A single arrow. It will completely dissociate into their corresponding products. But if it is a weak one, so we do have what? 2 arrows to indicate the compound is weak. Now, Ha is represented by a weak acid. Under solution, it dissociates what? Not completely. It dissociates partially to donate its proton, that is, it donates hydrogen ion, and the remaining compound becomes what? A minus. Now, A minus is considered as what? It's a conjugate base. It can accept what? Hydrogen to be able to form what? The weak acid. Now dissociation constant Ka is nothing but it is the concentration of the hydrogen. It is the concentration of hydrogen multiplied by its conjugated base divided by what? The undissociated weak acids. That means the larger the K means what? The stronger the acid. High amount of conjugated base and hydrogen can be what? Formed. The larger the K means what? The stronger the acid. But if you consider, for example, concentration of hydrogen, if you consider, for example, concentration of hydrogen and that of pH, as concentration of hydrogen is increasing, pH becomes what? Decreasing. Okay? The pH becomes decreasing. As concentration of hydrogen is decreasing, pH becomes what? Increasing. The relationship is inversely proportional. Just like that, just like concentration of hydrogen and pH, if you look Ka and pKa value, as Ka is increasing, pKa is what? Decreasing. As Ka is increasing, pKa is what? Decreasing. And as Ka is increasing, the pKa is increasing. That means the larger the K, the stronger the acid. Or in other versions, the smaller the pKa is, the larger the K. The smaller the pKa is, the larger the K, which is the stronger the acid. The larger pKa means what? The larger pKa means the smaller K. The smaller the K, the larger pKa. The acid is weaker ones. Now, what is a buffer? What's a buffer? A buffer is nothing but it's a mixture of undissociated acid and its conjugated base. A buffer is nothing but it is a mixture of solution which is a mixture of undissociated, undissolved acid and its conjugated base. And a buffer is what? It resists change in pH when an acid or base is added on a solution. And how a buffer is a good buffer? to say a buffer is a good buffer, we do have two criteria. One is the pK. The pH of the solution should be what? Plus or minus the pK of the acid. Okay. The pK must be near to what? The pH of the solution by plus or minus 1. If the pK is near to the pH of the solution, that buffer is a good buffer. And the other is the buffer should have what? Enough concentration of the undissociated acid and its conjugated base. That means concentration and the pK near to the pH is crucial to say a buffer is a good buffer. Now, according to Henderson-Hasselbalch equations, pH is equal to pK plus logarithm of unconjugated base and conjugated base divided by undifferentiated acids. From this equation, for example, if a weak acid is 50% dissociated, we will get what? We will get equal amount of conjugated base and undifferentiated weak acid. If these two amounts is equal, it becomes logarithm 1, and logarithm 1 is to mean what? 0. Then pH is equal to pK. That means you can see that a weak acid is 50% dissociated at pH equals to its pK value. But most of our metabolic carboxylic acids can have its pK value in between 2 up to 5. For example, if an acid has pK value 2, and if you compare an acid of a pK value of 5, which one is stronger the acid? The lower the pK, the stronger the acid. Now, a pK of 2 is stronger the acid than the corresponding acid which has pK value of 5. The stronger the acid means what? The acid loses what? Its proton easily. But the high pK value means the low K value, and the acid will have high affinity with the proton, and the acid will not release its protons easily. Okay. And as you know, the body produces what? More acids than base. Our body produces always more acids than base. Okay. We can get acids from the food what we eat, and the other is always we do have cellular metabolism, and this cellular metabolism produces what? Carbon dioxide. When this carbon dioxide reacts with water, it can able to form what? Carbonic acid, and this carbonic acid dissociates into what? Bicarbonate plus hydrogen ion. The larger carbon dioxide means the environment becomes what? Acidic. Acids produced by metabolism of lipids and proteins. Our body mainly produce what? Acids than base. And acids can be volatile acids, or it can be fixed or organic acids. Volatile acids like carbonic acid can be eliminated from our bodies through respiratory systems. Fixed acids like sulfuric acid, phosphoric acid, and the rest organic acids. The acid form, that is the hydrogen, can be eliminated from our bodies only through renal systems. Acids in our body, like organic acids like lactate, acetate, and pyruvate. Alkali, the main alkaline reserve for our bodies are bicarbonates. How our body able to maintain our pH value? We do have 3 lines of defense to regulate acid-base balance. The first line of defense is blood buffers, and Second line of defense are respiratory and renal mechanisms. What are blood buffers? Here, blood buffers will not remove the hydrogen atoms or the acids in our bodies. Rather, what they do is that they temporarily holds the acids. For example, if you do have water, okay, and if you put a sponge on it, the sponge absorbs what? The water molecules. Just like that, the blood buffers can absorb what? Hydrogen ions, if the condition is acidic. Okay, they will not eliminate hydrogen ions from our body. Rather, it will temporarily what? Hold on it. For example, if in our cells, if acids, for example, in the form of HCl, is added on it, if the hydrogen atoms came from HCl, that's a strong acid, blood buffers, what they did, it changed these acids into what? Weaker ones, and it changes to resist what? The pH change in a minimal way. What they did is that it exchanged strong acid or base for a weaker one, and results in much smaller pH change. What are the principal buffers in our body? In plasma, the main buffer system is carbonic acid, bicarbonate buffers. Whereas in red blood cells, the main buffer is hemoglobin. Okay, the main buffer in red blood cells are hemoglobin, whereas in plasma, the main buffer is carbonic acid, bicarbonate buffer system. Phosphate buffer also accounts 5% in red blood cells, whereas proteins also account 7% as buffer in plasma, like albumin. Albumin is a good plasma protein buffers because it is rich in histidine. Okay, it is rich in histidines. As you know, proteins are made from what? Proteins are made from thionein standard amino acids. Among these thionein standard amino acids, among them is histidine, and histidine has a good buffering capacity. Why? Because the pK of histidine is near to what? Near to plus or minus 1 of pH 7.4. That means the pK value of histidine is near to our physiological pH. That makes histidine is a good buffering capacity. And in general, those proteins which is rich in histidine can act as what? A buffering capacity. Like hemoglobin is rich in histidine, albumin rich in histidines. When you see bicarbonate buffer system, and this buffer is a predominant buffer in extracellular fluids. Okay. As I mentioned earlier, the main alkali reserve in our body is bicarbonate. The main alkali reserve in our body is bicarbonate, which is in the ratio of 20:1. This is the concentration of bicarbonate and carbonic acid. And bicarbonate is the main alkali reserve in our body. Since it is alkali, when the environment is acidic, it can absorb this hydrogen, it can react with hydrogen, and it can be changed into what? Carbonic acids. When this is action of bicarbonate, it can convert strong dissociable acids into weak non-dissociable acids and neutral salt without altering the pH of the environment. For example, if acid in the form of HCl is added on it, okay, the bicarbonate reacts with hydrogen and it changes into what? Carbonic acids. And this carbonic acid is nothing but weak non-dissociable acid, and later can be expelled through our lungs in the form of what? Carbon dioxide. Therefore, bicarbonate buffer is connected to respiratory system, and sometimes bicarbonate buffer is termed as respiratory buffer. And as the concentration of bicarbonate is high in our body will have what? Good buffering action through bicarbonate buffers. This graph indicates if a fixed acid or organic acid is added on carbonic acid buffer systems, the bicarbonate reacts with hydrogen and it will form what? Carbonic acid. The larger carbonic acid, it will shift into what? Carbon dioxide and water. And this carbon dioxide can be eliminated from our bodies through respiratory systems. The other buffer is phosphate buffer. Phosphate buffer is the major intracellular buffer. It is found in the kidneys, okay? And when hydrogen ions are added, these hydrogen ions can be fixed through alkaline phosphate, and later it is converted into acid phosphatase. And phosphate buffer is sometimes considered as a urine buffer. This is because what? These acids, for example, when hydrogen ions are added, this hydrogen reacts with the alkali phosphate and it changes into what? Acid phosphates. These acid phosphates can be excreted through our urine. As I mentioned earlier, our body produces what? More acids than base. And if you look at the pH of our urine, the average is around 6. Okay, pH of our urine is 6. That is acidic. That means our kidney tries to maintain our acid-base balance by eliminating what? Large amount of hydrogen ions. That makes our urine as acidic pH. Thus, phosphate buffer is connected to excretory systems, and phosphate buffer is also termed as urine buffers. The other is protein buffers. Okay, protein buffers are buffers or proteins which is found in the plasma as well as in the red blood cells. That means protein buffers are plasma proteins and hemoglobin. Among plasma proteins, albumin will account the highest percentage to act as a protein buffer, since albumin is rich in histidines. Hemoglobin is the best buffer for red blood cells. As you know, proteins are made from what? It is a polymer of amino acids. And an amino acid, from the name indicates, any amino acid, whatever it is, it has an amine plus an amine, which binds with the alpha carbon, and this alpha carbon also binds with the carboxylic end, the carboxylic atom. That's why it retains amino acids due to the presence of amine as well as carboxylic acid. Then, when two amino acids combine together through peptide bond, it will form dipeptide. It has one peptide bond, and we do have free amine group and free carboxyl end. If it is tripeptide, we do have two peptide bond, and still we do have what? One free amine and one free carboxyl. Therefore, proteins can act as a buffer. The carboxyl group gives what? Since carboxyl group is nothing but this COH. It can donate what? The hydrogen ions, what they have. Whereas the amine group, NH2, can accept what? Hydrogen atoms, and it becomes NH3. Therefore, by looking at their own environment, a protein can donate hydrogen ions or can accept the hydrogen ions due to the presence of carboxyl group and amine group. Protein buffers behaves as a buffer in both plasma and cells. In acidic medium, protein acts as a base, whereas in alkaline medium, protein can act as acid. And due to the presence of imidazole group of histidine, which is important in buffer role in proteins. Therefore, a protein which is rich in histidine can act as what? A good buffer. And albumin accounts greater protein buffer, which has 16 histidine residues. Hemoglobin is a major intracellular buffer in red cells, and a given hemoglobin contains 38 histidine residues. And hemoglobin also acts as a good buffer in red blood cells. The other mechanisms to regulate our acid-base balance is respiratory system. But respiratory system only works for volatile acids. A respiratory mechanism only works for volatile acids. It will not work for fixed acids like lactic acid, acetoacetate, hydroxybutyric, and so on. And body pH can be adjusted by respiratory system by changing rate and depth of breathing. And the other is kidney. Okay, kidney is the main organ to regulate acid-base balance. As I mentioned earlier, the pH of urine is normally acidic, around 6, but it may fluctuate, okay, based on your body pH. If your body is more alkaline, the pH of your acid shifts toward neutral pH or around 7.4 or like that. If your body is rich in alkaline conditions. Our kidney can control acid-base balance by eliminate large amount of hydrogen ions from our body. And when the condition of alkali reserve in our body, that is bicarbonate, is less, kidney tries to reabsorb what? Bicarbonate. And the other is kidney also eliminate large amount of what? Acids in the form of ammonium ions. Almost two-thirds of body acids or two-thirds of organic acids in our body is eliminated through excretion of in the form of ammonium ions through our kidney. If kidney fails, pH balance also what? Fails. And finally, this is the renal system, which tries to maintain our acid-base balance by reabsorption of bicarbonate ions, by excretion of hydrogen ions, by excretion of titrable acids in the form of what? Phosphates. Okay, and the other is by excretion of ammonium ions. And more than 50% of our organic acids can be eliminated from our body in the renal system through excretion of ammonium ions. Rate of correction, as you know, if there is any change in acid-base balance, blood buffers acts instantaneously, and then if it is volatile acids, respiratory system will come and tries to correct it, and it takes around some minutes up to hours. If not corrected, finally a renal mechanism will come, and it takes hours up to some days in order to correct acid-base balance. Can you hear me? Okay. Okay, let's finish it. That is non-covalent bonds. What is the role of non-covalent bonds in biological structures? As I mentioned earlier, when we see the level of organization of our cells, at the base, we do have what? Covalent bonds. For example, you know proteins. Proteins are macromolecules. How proteins are formed? Proteins are formed from 20 standard amino acids. One amino acid is combined together through peptide bond. It will form dipeptide. If it is 3, tripeptide. If it is 4, tetrapeptide, polypeptide, and then proteins. Now at the base, at primary level of organization, linear sequence of amino acids are combined together by peptide bond. And the peptide bond, it is an amide linkage, which is considered as covalent bond. Now, at the base, we do have what? Covalent bond, but as developed organizations are increasing to give more freedom for the flexibility of the macromolecules, non-covalent bonds are dominant. And non-covalent interactions are weak. That means it is reversible reactions, okay? Weak electrical bonds between molecules. Type of non-covalent interactions like electrostatic bonds, hydrogen bonds, van der Waals interactions. Now, the strength is what? It is very less when we compare with covalent bonds. But non-covalent bonds are very important in order to determine and stabilize the shape of biological macromolecules. And when it is individual, it is very weak, but collectively it becomes very strong. And these are some of covalent and non-covalent bonds. For example, ionic bonds, it's type of non-covalent bond which is formed by attraction of opposite charges. Hydrogen bond by sharing hydrogen atoms. Hydrophobic interactions. These are interaction of non-polar compounds. There is no attractive force between the two molecules. For example, when you see the lipid bilayer structure of plasma membrane, plasma membrane is mainly made by phospholipids, and this phospholipid has both polar as well as non-polar part. Now, the non-polar part wants to feel to contact with what? Water. And it becomes what? It becomes contact together in order to feel to contact with what? Water. But this non-polar part of phospholipids, in between them, there is no any attractive force. No attractive force in between them. But the driving force for their interactions what? Due to the properties of water. Okay. And the other is van der Waals force. This is whatever it is non-polar or polar, whatever it is, any molecules in which it becomes in close proximity. Now let's see one by one. First, let's see ionic interaction. This interaction occurs in between cations and anions, and this bond is non-directional. Unlike hydrogen bonding, ionic interaction is non-directional, and the strength of this interaction is dependent on the medium. If the medium or the solution is polar, the interaction becomes very less. The strength of ionic bond is less if the medium is polar. For example, if the medium is water, okay, the interaction of the ionic interaction becomes less in strength if the solution is what? Water. Because water has high dielectric constant, water is effective at screening of electrostatic interaction between dissolved ions. This is due to what? Water has high dielectric constant. For example, ionic compound like sodium chloride. Okay, when sodium chloride in water, it will undergoes what? So, as I mentioned earlier, now the medium is water. Okay, the medium was water, and according to second law of thermodynamics, there will have an increase in randomness of the system always, and largely responsible for easy dissolving sodium chloride in water. Now we do have sodium chloride. Okay, we do have sodium chloride in water. In water, it becomes dissolved and it will form what? Sodium plus and chloride ions. But the water molecules shields what? The ions and interact to each other. Now that means strength of ionic interaction is depends on the medium. Now, if the condition is now the medium is water, the strength of ionic interaction of sodium plus and chloride minus very less. Now, what is the function of ionic bonds or ionic interactions? Ionic interaction is very crucial, especially for determining the shape of mainly tertiary and quaternary structures of the protein. We do have 4 levels of organization of protein. primary structure, secondary structure, tertiary structure, and quaternary structure. Some form of proteins like myoglobin may not reach to quaternary structure, rather it will be maintained at tertiary level of organization. And the primary structure of a protein is made by peptide bond. If there is 16 molecules, there may have disulfide bond. If 16 amino acids are in close proximity, there may have disulfide bond at the primary level of organization. Whereas secondary structure is mainly formed by hydrogen bonding. Secondary structure of protein like alpha helix, beta plate sheet. Tertiary structure, it is the overall three-dimensional structures of the given protein, which is mainly made by non-covalent bonds. There may have some disulfide bonds. But that latter, if a protein, tertiary structure is the overall foldedness of a protein, but if this protein has more than one subunit, interaction of these subunits can make quaternary structures. And quaternary structures are mainly formed by non-covalent structures. It is ionic bonds. And ionic bond interaction is crucial for determining the shape of tertiary and quaternary structure of the protein. Ionic interaction also involved in the process of enzyme catalysis. When enzyme catalysis, ionic bond is crucial. Interaction also important for determining the shape of the chromosomes. And ionic interaction is crucial to stabilize polarized membrane of our neurons. The other is hydrogen bonds. Hydrogen bonds, as I mentioned earlier, hydrogen bond results from electron attraction between high electronegative atoms like oxygen and hydrogen atom that is bonded covalently to a second electron, like water molecules combined together through hydrogen bonds. And this bond is the primary responsible for water molecules interact to each other. Hence, hydrogen bonds belongs to non-covalent bonds. Individually, it is weak, but if it is too many, it becomes very strong. And these are some of the examples of important hydrogen bonds, which is found in different molecules. For example, hydrogen bonds in between an alcohol and water, hydrogen bond in between a ketone and water, hydrogen bond in between peptide groups. For example, secondary structures of a protein like alpha helix and the beta plate sheet is exclusively formed by hydrogen bonds. Now, as level of organization increasing, primary structure of a protein is made by peptide bond, that's covalent bond. If there's cysteine in close proximity, there may have disulfide bond. But as protein undergoes foldedness, the next bond, which is exclusively essential for the formation of secondary structure, is hydrogen bonds. And hydrogen bond also, it is very important for complementary basis of our DNA. Thymine and adenine combined together through hydrogen bonds. Guanine complementary base pair with that of cytosine through hydrogen bonds. And these are some of the function of hydrogen bonds. Source of unique properties of water. It is important for structure of proteins. It is important for DNA as well, polysaccharides, enzymes as well. The substrates can able to bind with the active site of the enzyme through the help of hydrogen bonds. And even a hormone, a ligand, can able to bind with the receptor through the form of hydrogen bonds. As you know, hormones can have every hormone can have their own different functions, but the function of the hormone is what? Only to ignite the receptor. Once the receptor is activated, the final response of the given hormone is totally dependent by the type of the receptor, not the hormones. The function of the hormone is only to ignite the receptor. Now, the hormone can able to bind with the receptor through the help of hydrogen bonds. And hydrogen bonds also crucial for matching messenger RNA to that of transfer RNA during protein synthesis. But as I mentioned earlier, ionic bond is non-directional. Okay, but here hydrogen bonds are as its directionality, and directionality leads to its strengths. The best alignment occurs when the orbital containing unshared electron pair of the acceptor, okay, atom in lines with what? The covalent bond between the donor atom of hydrogen. If it is in line, the stronger the hydrogen bond, and if it is bent, it becomes a weak hydrogen bond. Now, directionality is important in order to compare the bonding specificity by given Watson-Crick hydrogen bonds. Okay, directionality is crucial in order to attain to bind adenine with that of thymine, guanine with that of cytosine. The other is van der Waals interaction. Here, any atoms, whatever it is, okay, the molecule may be nonpolar or polar. Any atoms approach to each other, it will form electron clouds begin to overlap, and it creates an induced dipole. Now, this induced dipole, when the molecules are in close proximity and touching, it will form van der Waals interactions. What is the significance of van der Waals interaction? Individually, it is weak, easily broken, and it can be reversible, and it is universal. It can occur any atoms that near to each other, whether it's polar or non-polar. And it's important is that to determine strict complementary, and also it is useful for stacking DNA. Okay, DNA stacking is enhanced through the help of van der Waals force. And the final part of non-covalent interaction is hydrophobic effect or hydrophobic interaction. This refers to the association or folding of non-polar molecules in aqueous solution. Hydrophobic effect is its association of non-polar molecules in aqueous solution. Now, the aqueous solution, for example, in aqueous solution, as you know, it is a solvent is water. It's called aqueous solution. Is one of the main factors behind for protein folding, protein association, formation of lipid micelles, binding of steroid hormones to their receptors is due to their hydrophobic effect. And hydrophobic effect will not arise by some attractive force in between non-polar molecules. Rather, it is driven by what? It is driven by the entropy of water interaction. There is no any force of attraction between non-polar molecules. Rather, this non-polar molecules wants to feel contact with water. That makes these molecules in close proximity. And this refers entropy-driven aggregation of non-polar molecules in aqueous solution. This is not any attractive force, but rather it's thermodynamically driven force. And the hydrophobic effect is very crucial for the formation of plasma membranes. Also contributes for foldiness of proteins. When proteins undergoes foldiness, non-polar amino acids are buried themselves inside the protein, whereas polar amino acids are found on the outer part of the protein in order to contact with what? Water. Whereas the non-polar amino acids want to contact with water and very found on the inner part of the protein. Hydrophobic effect also is important for the formation of double helical DNA. These are the effect of hydrophobic. Here, the water molecule is highly ordered. Okay, this is the yellow part is non-polar substance. For example, if it is lipid, okay. Now here the water molecules occupies it, and it is in highly ordered manner. And thermodynamically, it's favorable to undergoes from lower entropy into higher entropy, and these non-polar molecules are in close together in order to feel to contact with what? Water, and water is released into the bulk in the solution, and the disorderness becomes what? increasing. That's why we say hydrophobic effect is thermodynamically favorable. Here we do have water molecules. This is a lipid. This is a lipid which is covered by what? Water. But all of them, all these 3 molecules of lipids wants to feel to contact with water, and to do so, it becomes what? In close proximity, and the water molecules become what? Released in disorder manners. This is my reference, okay? Now for this course, the best reference book for this module is Lippincott. You can read Lippincott if you do have time. Thank you very much. Thank you very much. If you do have questions, you're welcome. Samal? Hello?
ይሰማል።
እንቀጥል ወይስ ከሻይ
ትንሽ እንረፍ?
ትንሽ እንረፍ? እሺ። 5 ሰዓት ከስንት ላይ እንገናኝ?
አሁን 10 ጉዳይ ነው።
እሺ።
5 ከ5።
እሺ፣ እሺ።
5 ከ5 እንገናኝ።
እሺ።
እናመሰግናለን።